Good afternoon, everyone, and thank you for joining us today for the first of 3 virtual COVID-19 vaccine town halls presented to you by the Globe and Mail and the Royal Society of Canada. I'm Ivan Semeniuk, science reporter for the Globe and Mail. I'm pleased to be your MC and moderator for today's discussions. The Globe and Mail and the Royal Society of Canada have been collaborating for several months now on the Zero Canada Project. This is an initiative that seeks to direct us all toward the first day that Canada can report 0 new cases of COVID-19, something that we are all looking forward to. Our hope is that this series of town halls will contribute to the public good by providing information that Canadians need about the COVID-19 vaccines from leading experts in the fields of healthcare and science. So before we get started and introduce our expert speakers, here are a few housekeeping items that I've been asked to attend to. First, for viewers of our panel today, if you find you're experiencing technical difficulties, please let us know via the Ask box on your screen. And your questions will be answered as soon as possible. We have a technical team working. They're trying to ensure that everyone has the best technical experience we can provide. But in the event that we run into some problems, just bear with us. We'll try to respond to you promptly. Thank you to those who sent us questions ahead of time. We got lots and lots of questions. We'll do our best to get to them all, or at least hit the main topics that you're interested in. You can also ask a question during the panel by using the same Ask feature on your screen. We'll take a few of your queries closer to the end of the discussion. Now, everyone who registered for the webcast will receive a link to the recorded version so you can view the event again at your convenience. That's handy because there could be a lot of information today. We will also be sending links to further resources where you can find more information about COVID-19, including coverage from the Globe and Mail, information about the Zero Canada Project, and policy briefings from the Royal Society of Canada. The Royal Society has gathered more than 300 Canadian experts from across disciplines and across generations to produce independent analyses of the pandemic. You can take a look at the Royal Society resources at the link that you see on the screen. Hopefully you're seeing a link on your screen. I'd also like to take a moment to thank the organizations supporting today's webcast. They are the Social Sciences and Humanities Research Council, SSHRC, as many scientists know it, the Natural Sciences and Engineering Research Council, or NSERC, and the Canadian Institutes of Health Research. And if you haven't heard of these 3 organizations, they are the 3 organizations that fund much of the public research that is being done in Canada. Finally, if you'd like to know if you'd like to follow the social media conversation that's happening around today's town hall, just please follow at RSC, the Academies, all one word, for live tweets from today's event. And you can also follow that handle to stay up to date on news and events from the Royal Society of Canada, which is an umbrella organization for scientists across the country. You can also follow at The Globe Events for updates on upcoming events like this one from the Globe's events team. So now, without further ado, let's get to today's panel discussions. And I'll just say once again that this is the first of 3 panel discussions that the Globe is doing with the Royal Society of Canada, or offering to the public around, all focused around the topic of vaccines for COVID-19. Of course, we have 2 vaccines that have now been authorized in Canada, and there is the potential that we will have more. The Canadian government has made agreements to purchase up to 7 different types of vaccines. So there are at some point millions and millions of doses coming to this country, but there are many questions about them. The 3 town halls will cover various aspects of this process, including the rollout and what it will mean for people after they receive the vaccines. Today's conversation will focus more on the science of the vaccines, but there's certainly a lot to get into there. And to help us with this, we have our 3 panelists. And first, I will start by introducing first Dr. Michael Houghton. And as everyone should know, he is Canada's latest Nobel laureate. He's also the director of the Li Ka-shing Applied Virology Institute at the University of Alberta. He's a professor in the Department of Medical Microbiology and Immunology at the university there in Edmonton. Dr. Houghton is involved in developing vaccines against hepatitis C, Group A streptococcus, as well as therapeutics for Alzheimer's and non-alcoholic fatty liver disease, among other conditions. Previously, he worked as Vice President of Research for Novartis and Chiron Corporation in California, where he and colleagues discovered the hepatitis C virus, HCV, in 1989. And for that discovery, he was a co-recipient of the Nobel Prize in Medicine in 2020. So welcome, Dr. Houghton. It's good to see you.
Thank you.
Next, we welcome Dr. Alison McGeer, a professor at the University of Toronto and an infectious disease physician for Sinai Health System. I want to say Mount Sinai Hospital, it's the Sinai Health System. Dr. McGeer has served on international, national, and provincial immunization and infection prevention and control committees. Since February 2020, so not quite a year, her research team has been working on research funded by CIHR and the Canadian Immunity Task Force, focusing on the epidemiology and prevention of COVID-19. Hello there, Dr. McGeer.
Afternoon, Ivan.
Also with us today, Dr. Carolyn Quach-Tan, Chair of the National Advisory Committee on Immunization. Thank you for bearing with me. Lots of organization names. Of course, this is an organization that has been around for over 2 decades. Many Canadians are only finding out about it now that it exists, because one of the jobs of this panel, of this committee, is to make recommendations about how people should be prioritized for vaccinations. She's also a professor in the Department of Microbiology, Infectious Disease, and Immunology at the University of Montreal. Dr. Quach-Tan is a pediatric infectious disease consultant, medical microbiologist, and epidemiologist, and the medical lead of the Infection Prevention and Control Unit at CHU Saint-Justine. And please welcome Dr. Quach-Tan. Hello there.
Hi.
It's good to see you all. So we've got so many good questions right off the top. I'd like to first turn to Dr. McGeer, in a sense, to set the table. What some of our audience may not realize, Dr. McGeer, is how much you were involved in dealing with the SARS pandemic more than 15 years ago now, which of course was another coronavirus outbreak that was pretty scary at the time, but took a very different route to what happened with COVID-19. So I wonder if you could sort of contrast these two and tell us what it was like, as you've been working on the epidemiology of this now since last February. How has this version of coronavirus surprised us, and at what point did it become clear that really vaccines were going to have to be the end game here, not just public health measures as occurred for SARS-1?
Yeah, so, I think one of our problems with dealing with this COVID pandemic is that it's a coronavirus. When people looked at its initial genetic sequence, it looked like it had many similarities to SARS. And that made us think that it was going to be like SARS. Had we not thought that, we might have done actually a little better than we did in the initial response to it. SARS had a very specific epidemiology, which was relatively long incubation period, most infectious when you're really sick with it, really not infectious at all if you were asymptomatically infected or early on in illness. And what that means, those two things put together mean that you can do contact tracing because you have time from when people are exposed until they are infectious to other people. And secondly, it really focused it in hospitals, and that made it awful for a hospital experience at the time. But it allowed countries around the world, once they knew something about it, to control transmission and eradicate the virus. SARS-CoV-2, despite its similarity in name, is a completely different virus in its epidemiology. And what makes it uncontrollable, or made it uncontrollable enough to get us into trouble, is the fact that much of the transmission comes from people who are asymptomatic or very mildly symptomatic. Much of the transmission comes early in the disease. It's only got about a 4-day period from when you get exposed to it until you are infectious to other people. On average, really hard to do contact tracing that quickly, especially if you can't get test results back for 2 or 3 days. And so retrospectively, okay, if you look at some countries Taiwan, New Zealand, Australia, if you know the virus, you can in fact control transmission. So if we'd known all about SARS-CoV-2 when it started, we would have been okay, but we're not. And once a community transmitted virus is out there, once it is in sufficient quantity to get past our contact tracing ability, then our only solution is vaccines. That's a general truth about infectious diseases. They are what save us from important infectious diseases. And I think that issue became clear. I'm betting that the smartest people among us had figured that out by the second or third week in January, and most of the rest of us were a little bit slower to see that coming. But I think the people who've been working on vaccines were off and running second week of January, recognizing that there was likely to be a need for this vaccine.
Incredible, and that's a terrific setup. Of course, I have many questions for you again about the nature of the disease, but we will save that for a little bit later. Let me turn now to you, Dr. Houghton, because of course, you know, your professional life has been very much engaged in identifying viruses and producing vaccines. So, you know, when a situation like this arises, and of course, in many ways, this is an unprecedented situation, but certainly people have been anticipating and thinking about the occurrence of a global pandemic and how a vaccine response might occur. What are the key questions or features of the disease, you know, or what are the things that are needed in order to kind of initiate that process? And what's your sense of how the vaccine research and kind of vaccine makers of the world kind of pivoted to meet this challenge?
Yes. Well, I think viral vaccines usually work through neutralizing antibodies. Most examples of efficacious viral vaccines are the neutralizing antibodies that they induce. Those antibodies bind to the virus and prevent entry of the virus into host cells. It's also known that cellular immune responses also play a protective role, but I would say with most viral vaccines, most infectious episodes we've had, the key for vaccinologists is to make neutralizing antibodies. And if possible, cellular immune responses, T-cell responses. So, you know, we have lots of ways to do that. And some of them include growing the virus, purifying it, inactivating it, and then adding an adjuvant. There are many COVID vaccines like that have been manufactured in China. Other ways, more traditional, are to attenuate the virus in some way, where you can make deletions and put mutations in. That has been used for some viral infections in the past. And then more recently, a very tried and tested method is to make the surface proteins of the virus in mammalian cells, purify those surface proteins. In the COVID's case, it's the spike protein, and then add an adjuvant. And this method has been shown to be very efficacious for viruses like hepatitis B, papillomavirus, hepatitis A, and the shingles virus, varicella zoster. So we have all of these methods to use to make a COVID vaccine. They all take a fair bit of time, some longer than others, of course. But I think what we've seen is the new technologies of RNA vaccinology, as well as adenovirus vectored vaccinology. They've been able to come to the forefront because they have been able to be developed very fast. So I think the good news is we have lots of different ways to make vaccines to COVID. We've got the first ones out there, and time will tell which are the best in terms of potency and duration of immunity.
You know, just as a quick follow-up to that, you know, as you say, there are a number of different platforms. Some of them have really never been used before, or certainly not at this scale. We've got these two mRNA, messenger RNA vaccines that have been authorized. Health Canada is currently considering two adenovirus vaccines. And then down the road, there are some kind of protein-based vaccines that are coming as well. Are there specific advantages? You know, some are faster, obviously, to get to the regulators' table, but are there specific advantages that these different platforms offer, or how might it play out in the long run?
Mm-hmm. Well, they all are capable of generating neutralizing antibody, which is definitely what you need to combat viral infection. Some, for example, the RNA vaccines are also very good at stimulating cellular immunity. Some of the protein vaccines, for example, Hep B, primarily stimulate neutralizing antibody, not cytotoxic lymphocytes. So I think the new technologies are very good. The adeno vectored vaccine also stimulates both neutralizing antibodies and strong T cells. So I think these new technologies are looking very good, not only in efficacy, but also in the types of immune responses we want to elicit. But that said, we don't yet have a direct comparison of, for example, adjuvanted proteins with adeno or with RNA vaccines. We need to figure out which ones are eliciting the strongest immune responses, because the ones that do elicit the strongest are likely to be the most durable in terms of protective immunity.
Interesting. And I think in your answer there is something that many of us are beginning to learn. I feel like the whole world is getting an immunology lesson these days about how the immune system works in human beings, the fact that there's more than one mechanism for immunity, and that different vaccines may trigger some of these different responses. We'll come back to that, but now I'd like to get to Dr. Quashtan and just ask you about sort of how the world changed for you last year as chairperson of the committee for immunization, and considering that so many vaccines were in development, and by last summer we were beginning to hear that there were vaccines that Canada was making deals to purchase. I'm sure that started to seem like a call to action for you and your colleagues, because the question is, if these vaccines are coming, what does it mean for different populations of people, different ages, who's most affected by the virus in general, who might be vulnerable to other conditions or side effects? How do you begin to corral that question about how to get the vaccines to people in the best way possible?
Yeah, excellent question. I think we're hiring you, Ivan, in our committee. You understand it very well. So when we heard last summer that there were many vaccines coming, the first questions we asked was, we expect not to have enough vaccines to vaccinate everybody at first with the doses. And so we'll have to sort of prioritize who goes first. So to do that, we first, there were multiple things that were done. We first looked at literature review. So we hired the Alberta Research Group to do a systematic review to really pinpoint the risk factors for complicated disease. Because when we make a recommendation for a vaccine, the first thing we look at is burden of illness. So we want to know who is more at risk of hospitalization, of complications, and death. So that was their work. As part of that, we also needed to understand what the Canadian public wanted in a vaccine. So there were multiple surveys done with various Canadians to see who they would see as the most important group to protect. We also did that with various stakeholders in the world of vaccination, also to understand what their, how they would rank the various groups. We, of course, looked at what other jurisdictions were doing. So France was starting to rank their groups. The US was also starting to do that. So it was the UK. So we said, okay, so at least, we're not completely on a clean slate, but we know what other people are doing. And then what we did is look at the Canada's pandemic preparedness objectives, and which were to protect those most vulnerable, and 2, to limit societal disruptions. So putting all that in a box, we looked at the various populations we had. And so in the summer, NACI already had issued guidelines to say that we would start with the most vulnerable, being the elderly. So 70 and over was a risk factor that was the most associated with hospitalization and mortality, as shown by the ARCH group. And so that was our first group. We then went with healthcare workers, not because they were at more risk of complications, but because given reciprocity, these were people who were in the trenches taking care of patients with COVID, and we needed to protect them because we also needed them to protect the healthcare system, not only to treat COVID patients, but to also treat all the other patients who could be impacted by a healthcare system that would not be functioning. And then we added, the essential workers and the people with the underlying medical conditions, because again, in that systematic review, we saw that people with underlying pulmonary, heart, high blood pressure, and obesity were more at risk of complications. When we sent that out, we then heard that the first delivery of vaccine would only be 6 million doses, of both the Pfizer and the mRNA in the first quarter of 2021, which means 3 million Canadians that could be vaccinated. So we had to sub-prioritize the groups that we had already identified. And we also had to use numbers. So a group within the Public Health Agency of Canada with StatsCan then were able to, give us the numbers for each cohorts based on data they had on file. And that's how we came up with the second ranking, which was to say, start with people in long-term care facilities, because that's where the risk has been since the beginning of this pandemic, including healthcare workers taking care of them and their personal service workers. Then go to frontline healthcare workers, and that was remote communities or indigenous communities, and that was it for our 6 million doses. And now we're at a stage where we are ranking the rest of the population for stages 2 and 3, knowing that we are going to get 20 million doses in the second quarter, expecting to have more vaccines than just these 2 mRNA vaccines to see what can happen. And then you have to realize that in Canada, being Canada, health is a provincial jurisdiction. So that rollout is finally part of provinces' jurisdiction. And so the way groups are being prioritized within provinces might be a little bit different than what NACI had recommended, but that still is completely feasible. So at this point in time, I think that most of the provinces and territories have followed NACI's recommendations. And so that is where we're at this point in time.
Got it. And just one quick follow-up to that. One factor you didn't mention, and I know there are some questions about this, maybe this gets at the difference between immunity conferred by a vaccine versus what happens as an after effect of being infected. But should people who have already had COVID-19 be vaccinated, or is that a factor at all in considering the priorities?
So it's not a main factor. It's an important factor in the sense that what we know from people who did get the disease is that they are protected for a few months after disease. There are still reinfections. They seem to be rare. What is a little bit worrisome is that when people get reinfected, they don't necessarily elicit symptoms, but they might be able to shed the virus and therefore infect people around them. What we know from the phase 3 trials is that people with COVID that were confirmed by PCR in the past were excluded from trials, but people who had asymptomatic infections were still included. And so when you look at those two studies, you have approximately 10% of the population in the studies that had positive IgGs, which means that they had antibodies against SARS-CoV-2 at the beginning of this study. And these people had a similar vaccine efficacy because they were all analyzed together. So whether they put them in or they removed them, from the vaccine efficacy analysis, the point estimate was very similar. And they didn't seem to have more adverse events than people who did not have COVID before. However, in terms of equity, what NACI has said is that since we know that in the first 3 months after your infection, you're not likely to be catching another one, we would say that for equity reasons, wait, when you're called up, you can say that you'll wait for the 3 months so that people who are completely naive to this virus can be vaccinated first. Our group is currently doing a study of following up healthcare workers with COVID, and so that we're going to know eventually at one point in time they become susceptible again.
Got it. Excellent. Thank you for that detailed answer. And Dr. McGeer, so this actually sets us up for I think 3 other big questions that to some extent are related and may determine, you know, what the coming year or 2 will look like. The first question is, how much will a vaccine protect people against the disease? And that may be different for different vaccines. You know, but if something is 95% efficacious in a trial, what does that actually mean for people? Then there's a question about what's the difference between protection and transmission and what that might mean in terms of public health measures. And finally, how long will these vaccines last? Now, I know that there's a lot of information here that we don't know, but what are the potential scenarios that we might have going forward?
Okay, so let's start with, you know, what does it mean, how effective these vaccines are, and what does it do? So all we have at the moment is short-term outcomes after 2 mRNA vaccines and the first viral vectored vaccine from AstraZeneca. And with the 2 mRNA vaccines, in a very short period of time, post second dose of the vaccine, well, post first dose of the vaccine before the second dose, an 85% protection, which means that as with any vaccine, you can still get infected, but you are much less likely to get infected. Starting 2 weeks post second dose for the first couple of months, 94%, 95% protection with both of those vaccines. And it looks maybe, okay, in a hopeful sort of way, as if when you do get infected, you are less seriously ill. And that frequently comes with vaccines. And I think there's reason to hope that even if there are breakthrough infections in people who are vaccinated, that they will be less seriously ill. When those trials were set, our traditional beginning route with vaccines is we're focused on illness in the individual. So the first question you ask about a vaccine is, does it stop somebody from getting sick? The secondary question of, does it stop people from transmitting the disease to other people, is very much a secondary question. We would never recommend a vaccine to people for the sole purpose of protecting other people. We always recommend a vaccine if it's something that is a direct benefit to the person receiving vaccine. So we focus on that question. There was a lot of discussion when the vaccine trials were designed in the spring and summer about whether we were going to try to do trials that not only asked about protection from illness, but also asked about prevention of transmission. And though we eventually decided not to ask about prevention of transmission because it complicated the trials, it would have made them much larger. And people really wanted to, I think, to stay focused on, does this prevent illness? How can we get vaccine to people as quickly as possible? What that means is we don't have data on reduction of transmission. It is a general truth that if you prevent infection, you stop transmission, right? If I don't ever get infected with a virus, then I cannot transmit it to you. But if it is theoretically possible that what these vaccines do is they reduce severity of illness, but they don't completely prevent asymptomatic illness, and that asymptomatic illness might be transmissible. I think experts are somewhat divided on how likely that is. But given the severity of the pandemic and what's going on, I think we've all agreed that until we know that preventing illness in people is associated with prevention of transmission, we're not going to change our public health approaches to protecting people from transmission. It's going to be, I think, weeks before we have the beginning of information on prevention of transmission, probably months before we have really good data. So that's part and parcel of, this is going to be a long, slow transmission. Our vaccines are going to go up. We're going to learn a lot more about prevention of transmission. We're slowly going to be able to walk our way down. But it's a piece of the reason why it's going to take so long for us to get out of the public health measures we currently have in place.
Got it. And just lastly about duration of the vaccine. I mean, again, I know we're still learning here. You know, we've only had the virus in the human population for a little over a year. But how will we find out, and what will it mean?
So we're gonna find out by watching. You know, we will every day that goes by, there's another chunk of information that comes in about how often, what the risk of reinfection is in people who've been infected, what the risk of infection is in people who've been vaccinated. Fortunately, lots and lots of people following that, but you just can't, you know, when the virus has only been in the world for a year, you just can't say whether what your risk of reinfection is 2 or 3 years down the road, right? Don't have the data, can't do it. Right. Either way, you know, vaccines are still going to get us out of this, okay? The question is, I think, not whether vaccines are going to get us out of this, but which vaccines and how they're going to play out. It may be that we're moving into a world where coronaviruses change every year like flu viruses do, and you need an annual coronavirus vaccine. If that's true, we'll probably eventually have an annual coronavirus and flu vaccine, and you'll still get a single shot every year. It might be that we're, you know, just really lucky and the coronavirus does not mutate that quickly. I'm watching new variants. I'm less and less positive about this, but you know, it might be that its ability to change is in some way limited, and that we'll get really good protection in the long term, and you know, we won't need repeated vaccination. I think so. So there's two things about that. The first is that for those of us who are working on vaccines, for public health people charged with control, this is going to be a long, hard slog the next 4 or 5 years. But for all of us living in society, none of that downstream stuff really matters. Vaccines are still going to get us out of this. It's really, the only question is, you know, how fast and which vaccines. Got it.
And again, this is a good setup for the next question. I'm going to put this to Dr. Houghton, but then perhaps I'll invite the other panelists to weigh in after he's answered. You know, Dr. Houghton, this does sort of raise the possibility that we could be looking at periodic vaccinations down the road as, you know, the world kind of gradually tries to get its arms around this virus and other variants that may come up. Among the many questions that we've had ahead of the town hall are those that relate to vaccine safety. You know, and as someone who has set up trials and had to think about adverse effects and so on, I'd love to put some of those to you. I think very broadly, because of the coverage of the first vaccine, the Pfizer vaccine, and a few cases that have come up about allergic reactions, we have many questions from people about if they have allergies, you know, should they take the vaccine? And then also questions that are a little broader, people with different kinds of health complications, diabetes, heart, you know, other possible medical conditions, wondering about that as well. Now, I should say that Health Canada has some information about the directives for you know, or contraindications for who should take the vaccine. But, you know, maybe you could unpack a little bit of it for us in terms of your sense of safety issues with this vaccine and what that might mean going forward. Yes. With vaccines, I should say.
Well, I think the guidance is anyone that's had a history of severe allergic reaction needs to inform the clinician or the clinical nurses at the site of vaccination. I think, you know, there is a chance of getting a severe reaction if you have a history of severe allergy. Also, if you have a history of being allergic to any of the components in the RNA vaccine, this would be polyethylene glycol or polysorbate, then you are advised, certainly discuss it with your clinician, but certainly there needs to be a discussion on whether you should get the vaccine if you have a known allergy to any of the components. So the figures I've seen in the general community that don't have a history of severe allergy or and also that are not allergic to known components, the incidence of anaphylaxis, which is a severe allergic reaction, is around 1 in 100,000 vaccinees. So it's very rare. And I need to correct what I said. Many of those people that do have anaphylaxis do have a history of severe allergy. So in nearly all cases, it can be controlled. The individual might be hospitalized overnight, but it can be controlled. So the phase 3 clinical trial safety data was very good, I would say. It was rather typical of most vaccines. I had a shingles vaccine a few months ago, and that weekend I wasn't feeling very good. I had a fever, I had some malaise, and I was fatigued. In a way, that's all good. It means your body's reacting well to the vaccine components, and it's revving up your immune system. And often it's the activation of the immune system that makes us feel unwell, flu-like symptoms. So, so far, the vaccine seems to have a good safety record. And I think apart from the issue of allergy, it's looking good to me. You know, I think with the 2 RNA vaccines, they've gone into each 20,000 people during the phase 3 trial. And then now we've had a history of going into millions of people. Mm-hmm. And I think the main concern is people with a history of allergic reaction need to tell the clinical site they are prepared for anaphylaxis if it occurs.
It might be worth mentioning to people, and I don't know what your take would be on this, but because so many people, the goal, it really is to vaccinate everyone in the country, and of course that also carries on to everywhere else in the world. That's a massive vaccination effort, far beyond what is typical, even, say for a seasonal flu. Many more people will be vaccinated. So does that mean that we could still be in a situation where adverse effects could be very, very low, as a fraction of number of people vaccinated, but because we're trying to vaccinate so many people, anecdotally, we might see these reports popping up in the news more frequently than for other vaccines, if you know what I mean?
Yes. I mean, the annual flu vaccine is given to a good chunk of the population. I don't exactly know what it is, but I would imagine it's 30–40% of the population. So we already are good at doing that. In COVID's case, we need to immunize everybody. I think, we'll be looking carefully, public health will be looking carefully at the reactions after the second dose. And we'll be looking to see how long the immunity lasts. There is a distinct possibility an additional boost will have to be given after a year or 2 years. And once again, public health will look at any reactions to the additional boost. But I think so far, I think it's looking very good. The vaccine is looking efficacious after a few months. It's looking safe, apart from the allergy issue, which is always there with vaccines. And I think I'm very optimistic about being able to control the virus with these vaccines.
Got it. Excellent. I've got one more key question for Dr. Quach-Tan, and then I'm going to start to turn to some of our listener or viewer questions. But I think this is one that we should address, and I think you're the right person to take this up, Dr. Quach-Tan, which is we're currently in a situation of vaccine scarcity, which I think is exactly what you anticipated 6 months ago, to the point now where we are making changes or veering away from the manufacturer's recommendations on the time between the 2 doses for the Moderna and the Pfizer vaccines. So what can you tell us about the decision-making process and where the committee stands on whether there can be a stretch in the time between the 2 doses and how far that should go? Oh, Dr. Quach-Tan, you're muted.
Yes, sorry.
Yeah.
So what happened is that when we heard that we wouldn't have more doses than 6 million at first, discussing with the provinces, we realized that, we were caught between a rock and a hard place. So either we were able to vaccinate less people with the 2 doses and eventually even, keeping the second half of the doses allocated in the freezer to make sure that people would be able to have their second dose, or we were to say, based on ethics, equity, and efficient distribution of doses, we would say use all the doses you have to vaccinate more people with a first dose, and then aim to get your second dose within the interval as authorized by Health Canada. But if you can't, because your epidemiology in your province is such that you have transmission all over, it is possible to extend that interval. And what NACI said is that looking at the phase 3 trials where participants got their doses between 21 days and 42 to even 49 days, we said, well, at least we have some data to show that the efficacy that is provided as the average of all participants means that it's somebody between 21 days and 42 days. So we said preferably give that second dose within 42 days. Now, what people have a hard time grasping is that what we need to do is to try and protect the most people as possible, because it's now that the virus is being transmitted, that we are going to have a much more impact on hospitalizations and death and even cases if we're able to protect a good chunk of the population with a vaccine, with a one dose of a vaccine. So what we know is that in between those two doses, starting at day 14, as Alison said, you have close to 90% protection. What we don't know is when that protection finishes. So, we don't think that at 21 day, you're 90% protected, and on day 22, you're at 0. The same way we don't think that on day 42, let's say it decreased a little bit and you were at 80%, on day 43, you're at 0. It's a gradual decrease in your protection. For sure, we can't tell you exactly when you're going to have 0 protection. However, if you look at the timing of things, you could think that if you vaccinate more people now, given the seasonality of this virus that we've seen last year as well, given all the measures that have been put in place, come April, you should see a decrease in the number of cases, the same way we saw last year, and hoping that with the measures put in place and the vaccination, even if you have less protection, you're not going to be sick because you're not exposed to the virus. So that's I'd say the tension we have to look at between having a smaller number of people protected completely or having more of a public health protection so that your population is protected better. So we completely realize that it has raised tons of questions. Some people ask me, if I don't get my second dose, should I even get my first? My answer to them is, at least once you get your first one, you have some protection, and pretty decent one. I mean, 90% is way more than what we usually have with our influenza vaccine. In a good year, you have 40 to 50% protection. In a bad year, you have 0. We're talking about 90% protection. We know that it will last for a few weeks. We know that based on what we've seen with other vaccines given in adults with more than one dose, when the first dose is important because it primes your system, it gives you protection right away. The second dose is important because it allows your protection to mature and to be more stable and for longer duration. But there has never been an interval that's too long, so that if you don't give your second dose within, let's say, 6 months, and it was due at 1 month, even if you give it at 6 or at 12, usually your memory cells are still there and they're able to produce the protection that is needed at that point in time. There is something called a too short of an interval. If you give the 2 doses on day 1 and 2, it won't do anything because your immune system won't have time to have matured. But there's a possibility to extend that interval. So I think that what provinces have been doing were to say, we don't have enough doses for everybody. We want to be able to protect those that are more vulnerable. We're not going to vaccinate the entire population, but we would like to get, to the 70 and over or 60 and over, and protect our healthcare workers and protect our frontline first responders. That we would like to do, and we'd like to have a vaccine for all of them. And currently it's impossible, even more so with what Pfizer has told us last week, that next week there would not be any vaccine delivered. So I think, that's what really prompted the committee to make a recommendation that allowed provinces to stretch that interval a bit. If you don't need to do it, it's fine. But if you need to do it because you have, much transmission, then it's feasible. I might want to come back, just if you allow me, on the question of allergies, just to say that if you have allergies, even if you're anaphylactic to peanuts, for example, it's not a contraindication to vaccination. As Dr. Houghton said, what you want to do is to stay longer for observation at the site when you're vaccinated, but the only contraindication is allergy to PEG or to one of the components in the vaccine. Thank you.
Got it. This is where we're in kind of a suboptimal situation, and there's maybe some room for divergence of opinion. So I just want to ask Dr. Houghton if he has thoughts on this strategy as well.
Well, yeah, unfortunately, because we're limited by the amount of vaccine, public health have to be creative, and they are being. And I think what they're recommending is reasonable. The only doubt I have is if the delay of the second dose starts to get into 8, 10, 12 weeks, then my concern is that having looked at the neutralizing antibody response after the first dose, I see that the antibodies peak at approximately 2 weeks after the first dose, and by 4 weeks, they're already coming down. So then if you delay another 6 or 8 weeks, I'm very nervous about the level of protection that will be provided to those individuals. I think what NACI is recommending is to use the protocol, if you can, in the phase 3 trials. If you don't have enough vaccine, delay by 2 to 3 weeks. I think that's correct. But I'm just nervous if the delay is longer than that. If it has to be longer than that, then I tend to feel myself that we should give the vaccine doses on time to those most at risk, the people in elderly care homes and so forth. And then maybe the younger people who are still at risk for COVID disease, but at a much lower level, maybe we can delay the second dose for them. I totally appreciate what public health is doing, and what they're suggesting is very reasonable given the shortage of doses. But I start to get nervous when I see the second dose being delayed by 6, 8 weeks. Immunologically, I can see the antibodies are already very low, and the efficacy may be much lower than 90% if we delay that second dose too long.
Yeah, this gets us into a really interesting question about what we understand about correlates of protection, because post-infection, your neutralizing antibody levels also decrease fairly quickly, and yet out to 5 months you're protected. So we have this challenge of, if that's true of vaccines, then we're probably okay extending the interval for quite some time. If it's not, if vaccines are different and vaccines don't give you exposure to the whole virus, they only give you exposure to spike, then that decrease in neutralizing antibody might be more important. So I think I think public health people now faced with the choice of, for the next month or 6 weeks, I can provide 85% protection to 100 people, or 94% protection to 50 people. That's a really simple equation in the short term. But in the longer term, as Dr. Houghton has pointed out, we're going to need to be watching that really carefully, and trying to make sure that we're taking into consideration that potential loss and balancing it against what the immediate risk is. This is a really good reason, I would point out, to be living in Nova Scotia and Newfoundland, where the rates of infection are low enough that you can give the 2 doses on schedule and we'll be okay. Yeah. It's only those of us who are in provinces that are running, like Ontario is, 50 deaths a day, where that immediate imperative to try to get the death rate down and prevent people from becoming seriously ill is causing us to do different things.
Yeah,
got it. One more question. Yes, Dr. Houghton.
Yeah, no, I appreciate exactly your rationale, Alison, and you're in a tight spot not having enough vaccine. But the only comment I would make is that the efficacy after the first dose is around 90%, 2 weeks to 4 weeks after the first dose, okay? We actually don't know what the efficacy is after that if a person only gets 1 dose. Where we have to extrapolate because of the emergency and the situation, and it's reasonable to assume efficacy will be significant and substantial, but the point is we don't know. And if that second dose is delayed until 12 weeks, for example, the UK is kind of advocating that. The point I'm making is that we actually don't know what the efficacy is between 4 weeks and 12 weeks after a single dose. It's an unknown. But, you know, I totally appreciate under the circumstances, public health has to be creative and do the best they can. But, you know, I don't know logistically if it'll work out. I'd rather have high risk people get the vaccine on week 0 and week 4, according to the manufacturer's regimen, and maybe delay the second dose in people less at risk, like young, healthy people with no cofactors, if that's possible. It may not be possible.
We don't have enough doses now to
right.
do that is the problem, right? So we don't have enough doses at the moment to cover even the high risk. Even we actually don't have enough doses to cover even the highest risk of the high risk people. That's our fundamental problem.
Yeah.
And I think what is going to be key is to have good surveillance systems so that vaccine effectiveness is monitored as we move forward. I think we're all looking at Public Health England to see what their data are going to look like, because they are giving their second dose, as you were saying, 12 weeks after the first with Pfizer. So they're going to be the first ones to have data, we expect. And so we're going to be following that. But in Canada, we need to do the same. So whoever extends the interval needs to have a good surveillance system in place to monitor.
Yes.
Unfortunately, the UK data are brilliant. So that, you know, one of the good news stories is how well the UK are doing getting data out on all sorts of things, and so it's nice that that we can count on them to be able to get us that.
I really want to thank all of you for weighing in on this, and I think it's given our audience also a little bit of a glimpse, because of course we're all getting used to you know, kind of the regimented messaging that comes, you know, and sometimes with good reason. But everyone kind of from the same hymn book and so on. But, you know, in reality, there's incomplete information. We are awaiting more data all the time, and science works by constantly evaluating the evidence. So I think it's very useful for people to sort of see that discussion in progress, because you're all learning along with all of us. I think that's the real lesson here. I have a few more questions. I want to try to direct it to some that are coming in from our audience, and with the time that's left, kind of put them to each of you in turn. So you know, I'll give you one question, kind of have you each weigh in quickly on this. The first has to do with the next round of vaccines, or we're anticipating more. The first 2 that have been authorized are mRNA vaccines. They have a particular platform. There are 2 more in the pipeline that are currently being reviewed by Health Canada, both adenovirus vaccines, one from AstraZeneca, one from Johnson & Johnson. We don't know yet if they'll be authorized, but they're kind of heading in that direction so far, and there are results to come. How might that change the game in terms of, you know, what these vaccines could do? Might that change the prioritization? Might it bring new worries? Or even might people start vaccine shopping? Is that even possible if we have different kinds? I'll start with you, Dr. Quach-Tan. What happens if 2 very different vaccines then come on to join the 2 we've got now?
Yeah, so what's interesting, or more a challenge with the 2 new ones, at least with the AstraZeneca, is that the average efficacy is 70% compared to 95% for the first 2 that we already have, the mRNAs. So how are we going to, you know, reconciliate having a vaccine with a little bit of a lower efficacy, which is not 0, but is still lower than the ones that we have? So the committee is currently weighing on that to see how and if we use it, depending on if Health Canada is going to authorize it or not. The interesting aspect with the Johnson & Johnson is that in theory, it would be a one-dose vaccine. So it would be the only one so far for which, we could say, that's it, you have one dose and it's finished. Whether or not people are going to be able to shop for them, I doubt it. I think that provinces are going to have clear groups in whom we're going to use one versus the other, because all of these vaccines are provided through public health. It's not as if you can go to your physician and say, I'm going to pay for it. It's going to be whatever you get, where you get vaccinated. I think we still have to see the results from those phase 3 trials to see exactly what they really have in under their hood. But I'd say the next complication is when you have vaccines that have varied levels of efficacy, how do you deploy them? And for that, so that people do not feel that they are cheated because they're getting, let's say, the AstraZeneca, which has a little bit of a lower efficacy, when they know that if they wait, 6 months, they could get an mRNA vaccine.
Got it. And what did you mean by, in theory, the Johnson & Johnson vaccine is one dose?
So the data that have been submitted is for a one dose, but they've started in November, I think, a trial for a 2-dose series. So not sure what that is how it's going to end up, is if they're going to have a one dose authorized and then say later on, but if you want more protection, you can add that second dose. So there is this uncertainty that is still there. But what the data that have been submitted to Health Canada are with the one dose trial.
Got it. Dr. Houghton, I'm going to ask you to add to that, but also in the interest of time, I'll squeeze in a little bit more. I know a number of readers and today, but also before, and what we see on social media, people are concerned about vaccines that contain RNA and DNA instead of just proteins, and wondering if that has any impact on the body's genetic code. If you've probably followed some of that conversation, can you just kind of clarify what it means to have vaccines that are putting, kind of genetic information into our bodies?
Mm-hmm. Well, I don't think there's a risk with RNA vaccines. There is a small risk with DNA vaccines. We don't have them available yet for COVID, and we may not have them available. We'll have to wait and see. DNA can integrate into the human genome at a very low level and cause mutations and aberrant gene regulation. So there is a small risk with DNA vaccines, but you don't have to worry, in my opinion, about RNA vaccines. And the adeno-based vaccines, they're very safe. The adenovirus, when it's the wild type, causes colds, mild colds. But what Oxford and J&J have done is to delete various genes in the adeno so that it cannot replicate. It will not cause a cold. And what it's doing is it's delivering the spike protein very efficiently to the immune system. So I have no worries whatsoever about RNA vaccines or adeno-based vaccines.
Got it. And Dr. McGeer, do you want to add to that conversation about when we have other vaccines? People are also asking what will happen if they might be in a situation where they have to mix and match, or can they mix and match these vaccines?
So, I think at an individual level, none of us are probably going to have much choice about which vaccine is recommended. The good news about the AstraZeneca vaccine is, I think, there's a admittedly very small, but still a group of people who are allergic to polyethylene glycol. And so having another vaccine that people who are allergic can get will be really useful for that small group. But I think we're going to have to, in the same way that we've been listening to our public health authorities and doing what they ask of us to get through this pandemic, I think we all need to take a deep breath and say, that's what I'm going to do about vaccines. Remembering that a piece of getting vaccinated is that it's at the population level. So if we all get vaccinated with a vaccine that's 75% effective, then together the population has a much higher protection than that because of the concept of herd immunity. So I think we need to, I think, there's a lot of anxiety about when am I getting my second dose, and is this vaccine not quite as good, and what am I going to have in the choice about vaccines? And I think we need to be able to take a deep breath listen to what public health people are saying to us and get vaccinated with whatever is offered to us as soon as we can get it, because with all of that, we will get through this together. It's hard enough living in the pandemic without driving yourself crazy, worrying about whether public health people are making the right recommendations for you, getting vaccine. You know, as you've seen, there's a range. Nothing in the pandemic is perfect, but clearly vaccines are going to be much better than no vaccines. And that's why we have public health experts, so that we listen to them and do what we're told.
You know, we're just down to the last 2 minutes, so I'm just going to wrap up with you, Dr. McGeer, and maybe our other panelists will have a quick word on this. But when we get to the point where we're beyond scarcity for the priority groups and more of the general population can be vaccinated, I suspect there will be lots of people out there who are thinking, well, you know, I'm not living in long-term care, you know, I'm under 50 perhaps, I'm not really, you know, what are the odds that I'm going to have a severe case? Do I really need to get vaccinated? So what's your advice for people who might be making that equation?
So I think at the moment, certainly for adults, you know, the decision that you're making about a vaccine is about whether the benefit of the vaccine is greater or lesser than the benefit of not having the vaccine, or you can switch it in the reverse. And even for young, healthy adults, COVID is not a trivial infection. Yes, the great majority of people get better from it, recover from it completely, doesn't go on. But there are significant numbers of COVID long haulers. There's long-term loss of taste and smell, which is really miserable. And there are young people who get catastrophic illness and die from it, even though that's rare. So if you look at that and say, am I better off to be protected by vaccination and take a 1 in 100,000 risk of an allergic reaction, serious allergic reaction, or better off to leave myself exposed to COVID? You're definitely better off to get the vaccine. And that's I think the decision that I'd like people to be sitting thinking about, not whether COVID is all that bad, but whether getting the vaccine is better for me and safer for me than not getting the vaccine. And then there's a secondary question, which we don't have an answer to yet, okay? But hopefully we're going to demonstrate that these vaccines will protect other people, and then it'll be a little bit like flu vaccine. You're making the decision for yourself about prevention of flu, which you might not care all that much about, but you'll also be making a decision about whether you're protecting your friends and relations and colleagues from transmission. So a piece of it is we need to know more in order to let that young, healthy group of people make for what for them is going to be the best decision about getting vaccine. It's almost certainly going to be getting vaccine, but we'll have a lot more information by the time we get to them to help them with understanding that and making that decision.
Got it. Thank you, Dr. McGeer. And just quick last words, Dr. Quachtan, from you first, any final thoughts on the vaccine process?
So I think it's all fun and dandy to have all those vaccines. If people do not get vaccinated, it's not going to lead us anywhere further. As Alison said, to get out of this pandemic, we need the vaccines. And so NACI is going to continue monitoring the evidence, and we are going to adapt it as new data come up.
Terrific. And Dr. Houghton, your thoughts?
These COVID vaccines are efficacious, they're safe, and everyone should be vaccinated, in my opinion. You may not get sick, but you could easily pass it on to someone that could get very sick or die. So all of us have a civic duty to get vaccinated.
Excellent. Thank you very much. And I want to also thank the audience for all of the questions that you have sent in. Of course, there are many that we couldn't get to, but we did our best to get to as many as possible. The good news is, of course, there are 2 more town halls coming which will be, of course, oriented further to questions about the rollout and the implications of the vaccines. So please stay tuned for those, because there will be more opportunities and probably by then more questions to ask. This is the point where I would be asking all of you to give our panelists a warm round of applause for their time and their expertise and the thoughts they've shared with us today. I know we can't hear that applause, but I'm sure people are very grateful to have had this excellent information. So unfortunately, because of time, we have to leave it there. So on behalf of the Globe and Mail and the Royal Society of Canada, I'd like to thank all of the panelists for joining us. Thank you also to our supporters, SSHRC, NSERC, and CIHR. And a reminder to the audience that you will receive a link to the recorded webcast. That's the bonus. And thank you to all of our audience members again for joining us today. I'm Ivan Semeniuk for the Globe and Mail. We'll see you next time. Thanks again.